Hello and welcome to the NanoReach Podcast, a series where we learn about the lives and research of the members of the Nanomedicine Research Group or the NRG at the Institute of Chemical Technology, Mumbai. Our guest today is Dr. Aswani Kancherla, who is a research scientist at the NRG, overlooking projects that involve biophysical and structural characterization of peptide and protein therapeutics. He obtained his PhD from the Indian Institute of Science, after which he went on to the Johns Hopkins University School of Medicine as a postdoctoral researcher. This was followed by a brief stint of working in the industry at Minvax, and then he came back to academia and joined the NRG as a research scientist. Tune into his conversation with Sanjana Krishnakumar to know more about his journey in science.
Hey, so good morning. How are you doing?
I am fine, Sanjana. Yeah, thank you.
So, Dr. Aswani is a research scientist with our group. He works along with our PhD students. So, can you tell us about your association with our group and what is it that you do with the NRG?
Hi, Sanjana. Good morning once again, and thanks for this opportunity. So, my association with NRG happened sort of serendipitously. I relocated to Mumbai last year around this time, 2022 May, after completing postdoc and so on. I was taking a break and I was just relaxing. My wife also had her summer vacation, so we were traveling. We traveled to Northeast, which we had been wanting to do. So I came to know, and I hadn't yet started applying for any jobs. I was taking a break, and as you know, when you travel, you talk to the co-travelers, and I came to know that there is a position open at NRG through one of our co-travelers whom we met. So he's a gentleman, a professor from a university in Kolhapur. So he told me, "Oh, your skills match exactly with what they're looking for. So why don't you give it a shot? It's close to your home." So I didn't know about NRG then. I sent an application, and a few days later, Dr. Ratnesh Jain contacted me. And then I said, okay, can we meet so that we know each other's expectations? So I checked a little bit on the web, as anybody would do, and I found that we now are doing really good work, the two independent lines of work. And I saw myself fitting in one of the branches of the work that's happening here. Still, but when you want to join a place, you want to know how the atmosphere is. So I came, I met Dr. Ratnesh Jain in his cabin on a Saturday. We spoke for a while, and then Presha was there at that time. So she took me around the laboratories, and I met with some of the students. I saw the instruments, and then I really felt that yes, I will be able to contribute here. Then of course there was a formal interview and so on. And I joined last year in August, and now I finished about 10 months. Yeah, it has been going great. So my expertise is in structural biology, more biophysics, a little broader, and more specifically NMR spectroscopy. So we don't do much NMR spectroscopy here, but I have been contributing in these biophysical parts of the project. As you know, ICT has a close collaboration with industries. So Dr. Ratnesh Jain's group gets, our group now, so gets a lot of requests from industries to analyze their samples biophysically, and this is where I'm playing a major role.
So, how did you get into this area of biophysics and NMR spectroscopy?
One is as the things go with the flow, and of course I also had interest in structural biology. As a child, I liked almost all the subjects, and the first time I came across structural biology was during my undergrad. I did a BTech in biotechnology. So we had a course called protein engineering, where we learned about protein structure, how proteins work, and how they can be engineered to do what we want. And as you know, proteins are the workhorses of living organisms. So there was this topic where how proteases, serine proteases for example, act and cleave other proteins. There's a binding site, there's an active site. So active site is the place where actually the cleavage of the peptide bond happens, but the binding site is the place where you can tinker with and change the specificity. So you can engineer them to cut it where you want, say not at an arginine but at a lysine, or not at an aspartate but at a glutamate, by modifying the binding pocket. So it really captured my imagination, and I was fortunate to have association with some of the good research institutes. So I had a summer fellowship was in the Indian Institute of Science, and I had opportunity to look at the kind of research. So then I felt that I'll be able to contribute to this part. So after my BTech is completed, I joined for a PhD at the Indian Institute of Science. Yeah.
What advice would you give to students who are, in their studies, like in their bachelor's and master's, to make more connections in the field that they're interested in while they're still studying? How was it that you also, had all these connections?
So I completely agree. Connections are very important. I think a lot depends on the atmosphere you are there. So I was at the Bangalore Institute of Technology, where students are very ambitious. My students, as in my friends, and they used to explore. And we had good teachers who told us about, okay, there are summer fellowships, you apply and you will be able to go and see in real what is happening, what is research about. So we keep our eyes open and based on our interest, pursue those interests and talk to teachers, take their advice, and this is how. So the first time actually that I came to know about research, real research, is when I was in class 11. This was a program by CSIR institutes where they would select class 10 students based on their performance and invite them to visit their labs and pay for their travel, their accommodation for a couple of days along with a parent or a teacher or a guardian. So when I was in class 11, I got this opportunity to visit CSIR labs in Hyderabad. So that was the first time I could really see what research life is at a glimpse. So my father and I went, and there were also other students. So we stayed for 2 days there were 3 research institutes: Center for Cellular and Molecular Biology and the neighboring sister institutes IICT and LBRI. So there I saw the different aspects of research. People trying to modify plants to make them better, people working on Drosophila to understand their sleep cycle, circadian rhythms, and so on. So this was the first time I knew that, okay, research is something that happens. And then using the same program, I could visit their lab as an undergraduate during my summer and spend a couple of weeks. And at that time, I actually got hands-on work. I was associated with a PhD student who was working, Meenal Vyas. So she was working on Neurospora crassa and understanding genetics. There's a mechanism called repeat-induced point mutation, RIP. Whenever a part of the genome, a large chunk, is repeated, the organism naturally causes mutations in that region. So it was not known how the organism is able to identify repeated DNA sequences and mutate them. So anyways, they were trying to understand, and I was involved in, say, introducing, making some genetic modifications, isolating, and so on. In this way, I always enjoyed being in the research environment, and I pursued that area.
That's very interesting. So you also did a PhD at IISc, right?
Yeah.
So what was that experience like?
PhD in IISc was a wonderful experience. I would say it is one of the best parts of my life. I didn't mention, but my association with IISc was as a summer fellow. So after I did a summer fellowship at Hyderabad, the subsequent year I was selected as a summer fellow in IISc. So I fell in love with the place. It's a beautiful campus, and I really don't have words to explain, but there's a charm in that place. And for my fellowship, I was working in a genetics laboratory with Professor Mahadevan. His group was working on understanding bacterial genetics, how bacteria evolve to utilize certain sugars. So, for example, cellobiose is a degradation product of cellulose which is widely available in nature, but E. coli cannot utilize it as a wild type, but it can undergo mutations spontaneously and utilize that sugar. So they were trying to understand what are these mutations and so on. So I fell in love with IISc. It was the end of my BTech, and I came to know that some departments in IISc offer PhD even with BTech. There is no need for post-graduation. So after I completed my BTech, I applied for a PhD and I got selected in the molecular biophysics unit which aligns with my interest, like I mentioned, of studying proteins. So molecular biophysics unit is a pioneering department for structural biology in India. And I worked with Professor Siddhartha P Sarma as a PhD student. Our group during PhD utilized nuclear magnetic resonance to understand how proteins and peptides do their job, to understand their structure, and from the structure try to understand their function. So my PhD project was involved in studying small peptide toxins called conotoxins. It's very interesting. So there are snails, marine cone snails, which are sedentary. So they can't move fast. They just wait at a place. And they're carnivores. So they need to hunt and eat. Now, how would you imagine for a slow-moving snail to hunt something? Like all the fish are swimming very fast. So these snails hide, and when a fish swims by, so the snail actually ambushes and suddenly strikes and injects venom. The venom is so potent that fish gets paralyzed like in a fraction of a second. People came to know about this because there were a few fatalities, humans who were struck by these snails and who died or had extremely painful experience.
The people who were stung by the...
Yes, for example, people who snorkel, I think it was some cases were there in Australia. These snails also look very beautiful, cone snails. They have beautiful patterns, so you'll be curious and you'll hold it. So when you hold it, the snail may get...
It might think that you are a predator.
Yes, so it gets disturbed and it tries to protect. Either as a self-defense, it might try to envenomate you. So that's how it happens. So it's a few decades old research now. And people have identified that the venom contains a cocktail of peptides, several, about hundreds of peptides, short peptides, 10 residues up to 40 residues. And these peptides act in a sort of synergy to act on nervous system, muscular system, physiology, everything. And because of that combined effect, the action is so fast. So literally in a fraction of a second, you can find videos on the YouTube that fish get paralyzed and then it engulfs it. But why are we interested? Of course, as a basic science, we want to understand how these peptides are acting and so on. But as an applied part, when you purify the different components, some of those peptides can be used as therapeutics. For example, there is a peptide which is used as a painkiller for chronic neuropathic pain. It's approved by FDA. It's called zinconotide. So it's derived from these conotoxins. So our lab was trying to understand what is the structure of these peptides, how are they so specific, how do they act so fast. So these peptides contain 10 amino acids, but they also undergo several post-translational modifications like hydroxylation of proline, C-terminal amidation, then gamma carboxylation of glutamate, and bromination of tryptophan. But one of the important post-translational modifications is disulfide bond formation. So these peptides have high preponderance of cysteines, which has an SH group, as you might know, and when the amino acid sequence folds back, there is a covalent bond formed between the cysteines. So it will give strength to the structure of the protein. Different classes of conotoxins are known to have different disulfide pairing patterns. So my PhD project involved taking a conotoxin sequence which has several cysteines, 6 cysteines to be precise, and to identify its structure, what is the disulfide connectivity, and so on. So we took a peptide that was identified by another colleague by RNA sequencing, and we recombinantly expressed it. And I found that when you express it and purify this protein, it's so stable. You boil it, heat it up to 95 degrees, nothing happens to the structure. You put proteases, there are sites for trypsin, chymotrypsin on the sequence, but none of the proteases can cleave it. Why is it so strong? Why is it so stable? What confers its stability? So I labeled it with NMR sensitive isotopes and acquired NMR data and identified the structure. But this disulfide pairing forms a major component of the structure. So when I determined about 80% of the structure by NMR, it became clear that it forms like a sandwich, and 4 of the cysteines face inside of the sandwich as they are close by. So what is the disulfide pairing? So anyway, I already spoke quite a bit to cut the long story short. So I combined NMR, mass spectrometry, various tools to identify the disulfide pairing and the structure to understand what gives it the stability. So during this time, my PhD advisor has been extremely supportive. So I took long to figure out. So the disulfide pairing itself at the end of the PhD took about an year. I was struggling various methods. Mass spectrometry has been extremely supportive during all the time. And he's also very hands-on. He used to teach all the students how to set up these NMR experiments, which are very challenging. There are many variables to set to make it work. So he used to come with us, and I really had very good time. We still communicate frequently. So, as you said, PhD advisor makes a lot of impact on how you think about your research, how you think about even your life, so on. So, this study was published in ACS Chemical Biology, and it was also highlighted in Nature Chemical Biology.
Oh wow, that's amazing.
Anyway, PhD was a wonderful experience, yeah.
So after your PhD, I heard that you did a postdoc, right? So can you tell our audience what exactly it means to be a postdoctoral researcher, and what was your experience like as a postdoctoral researcher?
So, for many of us in India, after PhD, postdoc becomes a natural course of events, though it need not be necessarily the course of events. So, I wanted to go out and experience the differences in how they do the research and so on. The main difference between a postdoc and a PhD student, if you are asking me, the postdoc is expected to be more independent, come out with his or her own ideas, and mentor more junior colleagues like either PhD students or master students, and be of support to the advisor, the main PI, in grant writing. So essentially, postdoc is considered as a transition step. So for people who want to be in academia, set up their own labs, postdoc is considered a necessity so that they know the nuts and bolts of running a lab. So in my experience, I was at Johns Hopkins for 5 years as a postdoc. It was again a very good place. I really enjoyed. I continued to work on NMR and using NMR to study larger proteins compared to the peptides that I studied during PhD. Just I would like to add that many of the things that postdocs do, what I mentioned, mentorship, grant writing and so on, are for example already being carried out by PhD students at NRG.
Okay.
So our PhD students at NRG mentor master students or undergrads or new PhD students. PhD students here are also involved in some of the grant writing. So there is not a clear demarcation, but essentially postdoc experience is required for somebody who wants to continue in the academia and say maybe set up a lab.
So did you want to go into academia?
I am in academia now.
But was that your plan after your PhD?
Yes, I do like teaching and doing research, but academia is also very competitive. The main challenge most young investigators face these days is raising funds for the type of research that you want to do, publishing periodically to show that you're being productive. Yeah, this is something which I still fancy and which I consider. So, and which I'm able to do here as a part of NRG. I'm able to contribute in various activities, mentoring students, guiding them, planning the experiments.
You said that you worked in Bangalore for a couple of months before coming to Mumbai.
Yeah.
So what was that experience like?
So I completed postdoctoral fellowship in 2021 and I came back to India, and again I was at home just settling down, and I came to know that there was a position available at Minvax Private Limited. So Minvax is a startup founded by one of my graduate school professors, co-founded by one of my graduate school professor, Professor Raghavan Varadarajan. So this company is a startup now 7 to 8 years old, which works on vaccine design. So they were initially working on design of influenza vaccines, and when the COVID pandemic came up, they immediately used their expertise to switch to making COVID vaccine. So I came to know that there was a position available, and I joined. So Bangalore is a place where I have spent a lot of time as a PhD student, and I know the prof, I like him, and working with Minvax was great. My expertise is in biophysics, is very useful. So the idea of Minvax is you design a protein-based vaccine which is stable and survive even room temperature. So many times the challenge with vaccines is that you need to transport them in cold chain. For example, the mRNA vaccine that we had for Moderna vaccine, I think for COVID, had to be transported at minus 20 or something like that, or liquid nitrogen temperature. So when you have countries where there are no cold chains available, or you want to transport them to remote areas, this becomes a challenge. Professor RV's RV is Raghavan Varadarajan. So his expertise is in understanding how to make stable variations of these proteins without losing their immunogenicity. So they are working on making these COVID vaccine, which can be transported, say for example, at 37 degrees Celsius, and it still works fine. So I was there briefly for 6 months, and I settled down, and I really had a very good time. Though it is a company, it was like a big lab, because it is a startup and it's a mostly R&D work. What is very different in an industry or a company setup is a very strict timelines, because they want to get things done. As a PhD student or a researcher, you're more focused on, okay, I want to understand what is happening in this question, what is the mechanism. You are more focused on the understanding and trying to figure out exactly what's happening. In an industry setup, while you want to understand, but there's also a timeline. So sometimes you want to coordinate these two, or most of the times you want to coordinate these two, and if timelines are more important in certain cases, then you will, okay, this is something which we cannot find out now, so let's have to move on. What is the plan B? So being in Minvax, I learned these aspects, that how to plan and stick to your timelines and so on. Unfortunately, I had to leave the place and relocate, because for various personal reasons, my wife and and her family is here, and so I decided, spoke to RV and said I have to make a move to Mumbai.
So one thing I was curious about after reading your profile is that you have experienced research in a startup, you experienced research at the undergrad level, you experienced research as a PhD, as a postdoc, and you also experienced research in India and outside India. So a lot of Indian students often aspire to go abroad for their PhDs or for their postdocs. So do you think there is a country-wise difference in the way people do research in India and outside India?
There are various aspects to this question. So this was one of the things about which I was curious when I was going to USA. I said, okay, what's the difference? What will be the difference? So whatever answer I give is dependent on just my experience. So you should always take it with this pinch of salt. I was at one of the best places in India, and even in USA it's one of the best places, Johns Hopkins. So both places are great. And if you ask, is there any particular difference the way, for example, research groups function? I would say no. I mean, the good labs in India are as good labs in Johns Hopkins, what I observed. If you ask how is the teaching quality, the profs who taught me in ISC are among the best, and I audited a few courses in Hopkins. They're also good. So these are all similarities. Students work together, there is a lab culture, there is a cohesion among groups. These similar. People hang out, so culture-wise. But what are the differences? So in India, one of the main differences that struck me when I went to the USA is speed with which you could get things done. This is probably logistics problem. For example, in India, I was working with the NMR machine, and say something happens to a part of the amplifier, which gives electromagnetic radiation to the sample. In order to get it repaired, you have to send it to, say, Germany. Then there is shipping process, they repair it, and it comes back. Timeline is 3 to 6 months. As soon as I went to the USA, there was an amplifier problem. I sent an email saying, hey, our amplifier seems to be out of order. I don't see the signal coming up. Can you please take a look? The next day, before I came to the lab, a spare amplifier already shipped.
Oh, wow.
So this is because the facility is there. There is no import, export, customs. It's fast. So that becomes a major advantage, for example, if you are in the West. But I think things are changing now. Even in India, we can get things done very rapidly. So that's one of the main things, speed with which things get done, because of the location, because of the geography. That was one thing I found. Then the other difference that I found is being strict about following regulations like lab safety, having eye wash, discarding solvents, then doing courses about ethics, research ethics, reproducibility. So when I went to the USA, I had to do all these courses, write an exam, make sure that I understand, or to them prove that I could pass the exam. So I think again this is again changing in India. So I did PhD now several years ago, and I see as I was doing my PhD, things are changing in India also. And another thing which I would say is different is there's a lot of there's support for students in terms of their health care, mental health issues, and that sort of support I found quite a bit in the USA. That universities have, that there's a place if you have challenges, you're having anxiety or something like that. There's a place whom you can go and talk to. There's a therapist whom you can talk to. Support system is there. But for the third point, I would say that this is, I think, a consequence of the culture in the West. For example, that was so for the non-scientific part of the differences. When I was in India as a PhD student, there's a lot of friend circle hanging out. There is a mess service in the institute, so you go have your meals with other people. But there's something missing in the West when I went, and it's because of their culture, non-scientific culture. It's more individualistic. You come, do your work, and once the work hours are over, probably you're back for your own family and so on. So this is one thing which I found difficult, for example, when I went to the USA. So I had a lot of friend circle in India, and once you go there, you're mostly colleagues in the lab, and the culture is different. So this is one thing which I would usually tell people going to the West: be prepared. So I was informed about this, but even after knowing, you will, once we go there, it takes some time to settle down and figure out. But otherwise, if you ask about research, the good labs in India do as good research and as great research as good labs in the USA, for example, where I have seen. Yeah.
That's really interesting to know, actually. And I completely agree with the instrumentation and the repairs part, because I was talking to some of the PhD students when I joined about one of the more expensive instruments in our lab. I think it was the FTIR, and people were telling me that we have to pay several thousands of rupees for customs just to get it here. So I'm assuming those problems would be a little lesser, right?
Yeah. This is where being self-reliant in instrumentation, etc., makes a lot of difference. Since we are getting the instruments from abroad, these are a necessary evil of the system. You need to undergo customs, and but what you say is true.
So you have had, again, so many years of experience. So if you could say anything to your younger self, maybe your 11th standard self, before they discovered what research was like, what would you say to your younger self?
I don't know. I feel that I have been very fortunate. I don't know how things could have gone differently. I have had great teachers in school, college, great mentors during PhD, postdoc, even now. So I really feel fortunate. But maybe if I have to really tell myself to do something differently, maybe I would tell myself to set timelines and keep a track of time. So during PhD, there are times when I would just keep trying. It's not working. Okay, let me try something else. So just a balance. Just keep track of your time, make plans, and see. So to be able to tell when something has to be left alone. But as you would realize, these advices cannot be generic. Everybody has to retrospect and introspect and figure out what works for them. Yeah.
And what advice would you give to anyone who's starting their PhD now, or who want to do a PhD sometime in their careers?
Okay, this one also a tricky question. So I would say a few things, maybe what I found useful as I try to imbibe them. So one of the things is my PhD advisor used to tell me, Ashwani, take your work seriously, but don't take yourself seriously. So I think what this means is when you take your work seriously, you will give your full 100%, work with honesty, dedication, and full involvement. At the same time, don't take yourself so seriously so that you'll be able to crack a joke on yourself and maybe take life in a lighter vein, so it will help as you're working. And other thing I would say is coming from your previous question. I would tell the PhD students or anybody in the research to make plans, make timelines, keep a track. So even if you make incremental progress regularly, it will make a lot of difference over a course of time. So we may not make significant progress on a day, but if you are making a little by little every day over the course of time, it makes a lot of difference. So just make it a habit to give that little bit extra every day. So this is again something I came across somewhere, and I try to remind myself that okay, try to do a little bit more every day. An interesting analogy for this is if you take 0.99 to the power of 365 and you will get about 0.02, and you make 1.01 to the power of 365, you will get 37, and both are about 1500 times different. That means if you do just a little bit 1% more every day, at the end of the year you would have achieved about 1500 times more. So just make this, keep it in your mind. And the third point I would like to say is PhD and research is a difficult journey, but it's very fulfilling. You are trying to find something that is unknown, you are trying to contribute to the knowledge of the world. So remind yourself that it's not going to be easy. Experiments fail, but reminding ourselves that we are trying to contribute to the knowledge of the humanity will keep us going. And there are many other things also which in this journey we'll make associations, we'll make friendships, we'll learn a lot of soft skills. So all these things are going to help us, and most importantly we have to enjoy this journey. So this is what I would say. Remember, remind yourself it's very important and enjoy the journey.
So I follow a lot of science communicators who are often PhD students themselves, and they get so absorbed into their research that if something goes wrong in one of their experiments, it takes a very big toll on their mental health and their self-worth. So that's why a lot of them say that work-life balance is very important during any kind of research you're pursuing, whether it's a master's or a PhD or a postdoc. So what do you think about that? Do you agree with this kind of a statement? Or?
So I agree partly. What I feel is that experiments are supposed to fail because you're trying to find out something for which there is no standard operating procedure. So when experiments fail, we should realize that it's part of the game. It should not affect our self-worth. So I was yesterday reading an article on the importance of being stupid in sciences, or importance of stupidity in science. I'll share it with you. So I think here it's the mentor or the PI should let the student know that it's okay, it is natural that experiments fail. That's part of your question. About work-life balance and getting involved into the project, in my opinion, it is natural for you when you're trying to work deeply on a question to get engrossed in it. It's quite natural. But definitely, I agree with you that you should make sure to have time outside lab. You can maintain your both mental and physical health. So different things work for different people. For example, when I was doing PhD, I used to go and swim in the evenings. So that used to be very relaxing. Or there are opportunities to attend some concert or have a hobby, sketch. Do pursue your hobbies. Like if you as any student who know how to sketch, sing, dance, anything, do. And many times these go hand in hand. Sometimes students wrongly feel that it's not the productive time, but with experience you will realize that when you spend time on your hobbies and passions, you come back to work with more vigor, rejuvenated. And I would like to also share an anecdote that one of my PhD colleagues, she sketches, and it so happened that she had to work on a project from protein in the islands crystal. Once her paper got accepted in biochemistry and they wanted a graphical abstract for her paper, she actually sketched an eye and used it in her own paper. So even otherwise, I think what you said is correct. That it's very important to maintain both mental and physical health, because we should consider PhD or research more like a long marathon. It's not a short sprint. It's not that you can give your 110%, give a burst of energy and leave. it's going to be a sustained, prolonged effort. So it is important to maintain your mental and physical health, have activities outside the lab, and every individual has to decide what is the best way to spend time outside the lab.
That makes a lot of sense, and I also think that just pursuing a hobby, even if it's not in a PhD, along with any professional job that you're doing, it always just gives you a big mental health boost. That's very important to maintain. But yeah, sir, I really enjoyed talking to you this morning, and I hope you also enjoyed sharing your experiences with our audience. And yeah, thank you very much for coming and speaking with me today.
Thank you, Sanjana. Thank you very much. It was a pleasure talking to you as well. Thank you.
Thank you for tuning into this episode of the NanoReach Podcast. If you like this episode, please share it with your peers and subscribe to the Nanomedicine Research Group on YouTube and Spotify for more such episodes. Follow us on Instagram at nrg_ICT Mumbai for regular updates. Thank you.